The irradiation system is surprisingly simple. The electron beam power supply is capable of generating almost 1 million volts, used to accelerate electrons toward their target. A concrete enclosure houses the heart of the system, the electron accelerator and scanner. Behind the concrete shield, the electron accelerator directs a powerful beam of electrons through the scanner at a layer of sludge flowing beneath it. Sludge, or any other material that can be piped in from the treatment plant, is presented in a thin layer so that every bit of material is irradiated in a fraction of a second. The toughest problem researchers have had to solve here is to present sludge to the electron beam in a uniformly thin layer so that no part of the sludge escapes the minimum dose needed to kill viruses and other pathogens. This unit can irradiate 100,000 gallons of sludge a day, the amount produced by a city of 200,000 people. The 7-foot-thick concrete enclosure surrounding the radiation chamber absorbs X-rays produced when the beam hits a target. The absorbed energy from a disinfecting dose of 400,000 rads only raises the sludge temperature by 1 degree centigrade. Scientists from both MIT and the University of New Hampshire are involved in assessing the effectiveness of the electron treatment on bacteria, viruses, and other pathogens. Many key experiments begin with the addition of a known quantity of virus, usually polio, to the sludge flowing through the system. The intense electron beam causes the sludge to fluoresce. The high-energy electrons penetrate the sludge and accomplish their work in a fraction of a second.
We all set to go at 60 milliamps, Bruce?
Yes, Ron. We're stable at 60.
In each experiment, sludge samples are irradiated at several dose levels.
Okay, here's 50.
But I have 40 now, Bruce.
Going to 40.
Okay.
Finally, a sample that received no electron treatment at all is taken as a control, and the samples are transported to the laboratories of the University of New Hampshire. This is where all the virological studies in connection with the Deer Island project are done. Dr. Ted Metcalf is in charge of the research.
Our objective here is to determine how effective energized electron treatment is in killing enteric viruses. That is, viruses that pass through the human gut and that are found commonly in sludge. Viruses are incredibly potent packages of infection. So we need to make sure that any treatment that we give sludge is indeed effective in eliminating viruses as a health hazard.
Before virus content can be determined, viruses must first be recovered from the irradiated sludge samples in a special solution. Viruses are true intracellular parasites. They can reproduce only within living cells. For this reason, cultures of living cells are used in these experiments. A healthy, normal cell culture looks like this: a smooth, even layer of adjoining cells. To test for the presence of active viruses, the cell cultures are inoculated with the specially prepared samples. Each cell culture is then stained with a dye that makes living cells turn red. Colorless patches, or plaques, will form where the cells were infected with living viruses. After inoculation, the cultures are incubated to give viruses a chance to replicate. Several days later, the colorless plaques of infected dead cells are counted. The more viruses present in a given sample, the more plaques. From these experiments, a clear picture of the effectiveness of sludge irradiation has emerged. At zero irradiation, there was nearly 100% virus survival. As the radiation dose increased, the percentage of surviving viruses decreased in proportion. At a dose of 400,000 rads, no virus survived in digested sludge.
What we really should say is no virus survived that we can detect. Some viruses, like infectious hepatitis and gastroenteritis viruses, cannot be recovered routinely in cell culture systems. Nevertheless, everything we have learned to date indicates that these viruses will be killed by radiation, just like others that we can recover. What this means is we have a method here that will virtually eliminate viruses as well as other pathogens in sludge as public health problems.
Clearly, the treatment of sludges by energized electrons has the potential of being an economical and environmentally safe technique for disinfection when used as part of an integrated system of sludge management. Our previous concern was with possible danger to humans. But we might also ask, how does the soil ecosystem react to sludge application to land? If we elect to use land application for sludge management, its compatibility with the soil ecosystem is essential. Assessing this compatibility is one of several research projects being conducted at the Soil Invertebrate Laboratory of the College of Environmental Science and Forestry in Syracuse, New York. The work is under the direction of Dr. Roy Hartenstein.
Soil is a very complex ecosystem which is inhabited by numerous creatures, including a wide variety of earthworm species. Now that sludge application to land is being considered very seriously, we have to ask ourselves whether that application is going to do harm to the land and its inhabitants. And on the other hand, how can we use the soil organisms to manage the sludge most efficiently?
Part of the research was aimed at finding out whether earthworms survived when allowed to feed on different types of sludges and sludges mixed with soil. In one series of experiments, a carefully weighed quantity of earthworms was placed in dishes containing various sludges. The dishes were set aside to allow the worms to feed on the materials. After a time, the condition of the worms was checked. Early experiments showed that some sludges were very toxic to worms, killing them within hours. The toxic sludges had all been anaerobically digested. On the other hand, worms thrived on sludges that had been aerobically digested. As they passed the material through their bodies, large, tough clumps of sludge were broken up into castings, which are similar to topsoil. As a result of this laboratory research, a practical experiment to do this is now underway at the San Jose wastewater treatment plant in California. Dr. Hartenstein.
We've really just begun to learn how earthworms may be helpful to us in managing our very serious sludge problem. For example, we have strong evidence that the earthworm is very effective in reducing the number of salmonella and other pathogens that are present in sludge. Also, once sludge has passed through the animal's body, the castings which the animal has now produced are totally devoid of odor. What it amounts to is that we have many more options open to us to manage our sludge than we had previously thought.
With many treatment techniques available to the designer of wastewater treatment plants, and considering the wide range in wastewater composition, the question for the plant designer and operator becomes, which mix of options will work best and be most economical? This question is being investigated by Dr. Richard Dick and his associates, working first at the University of Delaware and now at Cornell University.
Present practices were developed by engineers with an urgent need to solve sludge management problems. The work we are doing differs from much of the work of the past in that we are basing design and process integration on the best fundamental understanding of process performance we can develop. There's a great need for improved economy and effectiveness in sludge management. Modern digital computers make it possible to take into account the complex interactions between processes so that we can evaluate how best performance and economy can be achieved. To illustrate how this works, let's consider a city of 75,000 people. They would produce a wastewater flow of about 10 million gallons a day. And let's treat that wastewater by primary sedimentation and the activated sludge process, and treat the sludges produced from primary and secondary treatment by gravity thickening to increase their concentration, anaerobic digestion to stabilize the sludge, and then apply it to agricultural land. Let's consider that it's trucked 20 miles. For this illustration, we have constrained the size of the gravity thickener to 1,000 square feet. This would be a rather traditional design value for a city of this size. The resulting cost of sludge management, including amortization of capital, is $366,000 per year. Now, let's tell the computer not to fix the size of that thickener by the traditional criteria, but rather to pick the size which gives least overall cost for sludge management. In this case, the size of the gravity thickener is 1,700 square feet. That's a 70% increase in the size of the thickener, and it's going to cost more. But that extra expenditure is more than compensated by the savings in the cost of other processes with which the thickener is integrated. In this case, the total annual cost for sludge management will be $250,000, or a savings of $116,000 each year. Results of this research can be useful to planners, consulting engineers, managers of wastewater treatment facilities by showing the implications of alternative sludge management approaches.
Until recently, putting sludge on land had been considered an undesirable and even unsafe practice from the public health standpoint. The viruses and other pathogens in sludge could make it a link in a chain by which disease might be transmitted from infected to healthy people. We now have the means to break this cycle of disease transmission and to reduce potential health risks. Thus, disinfection of sludges by energized electrons, followed by direct injection into soil, may provide us with an acceptable, technically superior, and more economical method for sludge management.